Thanks so much for the opportunity to give this talk. I'm going to try and give a pitch today of why, I'm coming up with this fancy name, a variant of unknown mechanism, but it's really why should we care about biophysics in this field and when we're doing these high throughput screens. And I mostly study membrane proteins because I've really fallen in love with them because they play the role molecularly to serve as the interface for the physical and chemical environment around the cell. And this molecular role allows us to have complex physiology and sensory physiology so we can see the world and interact with the world, but also so we can have electrophysiology so we can, think about other things when we're in talks or like our experiments or just like what we're going to eat later or something, But of course, membrane proteins, because they're at this interface, they also serve as the direct targets for most drugs or indirectly move solutes across membranes. And mutations within membrane proteins, of course, have really underlied their diversification, so we can have complex physiology, but also, mutations cause diseases that ail us or alter the efficacy of drugs we use to treat these diseases. However, there's many different mechanisms by which a mutation can have its effect, ranging from the biophysics of folding to the cell biology of trafficking, all the way to structure function mechanisms. I saw this really cool piece from Doug Fowler and Heidi, which is like, are we going to have this problem solved by 2030? Is the VUS problem? And we're thinking about VUSs a lot, and I enjoyed this piece a lot. I felt like it was relatively tempered and laid out a path of how we might do this, and I think we might get there. And if you ask some people at Google, they'll tell you that they've solved this problem recently, and we might not all agree with that, but they got a press release, so good for them. And I dream of the day when Debbie or Yoon Song or all these amazing computational people really put us out of a job. But I don't think we're there yet, unfortunately. And even though they and then Google will take credit for it, and it'll be really sad. We have this pathogenicity score that they created, which makes it sound like they've solved everything. But there's a lot of complexities in genetics. We've been hearing a lot about these, actually. I've really enjoyed the talks about thinking deeply about heterozygosity, or, we have problems like dominant negative effects that are in mutations where, like, you have an ion channel, and if you have a mutation in one monomer that faces the pore, it'll kill the whole channel, and you'll have problems. There's penetrance in interactions. I'm really just, as a biophysicist, mansplaining genetics to you all. So I'll stop doing that. I apologize. But, I think mechanism really matters. And one of the reasons mechanisms matter, and I think one of the best examples of this is CFTR. Many of you probably know about this story. But, the chloride channel CFTR causes cystic fibrosis, where mutations will break its function, and then you get accumulation of mucus, which causes disease. And there's been developed a bunch of different classes by the CF community for a long time to describe how mutations break CFTR. And I think it would be really useful for us to start to think about as a field, how do we create general classes that also explain how mutations break things? Because you can look at CFTR and you find that, say, the majority mechanism by which mutations break CFTR is through maturation processing, what they're calling, or I think about biogenesis, so whether the protein forms or not. And this information was really useful when Vertex first started trying to find drugs for this, where they started with one mutation in drug screens, this glycine 5551 aspartate, where they used that to find potentiators or channel openers drugs. And then they switched to the delta 508 mutation, which is in most patients, and they were able to screen based on that to start to find correctors or ones that rescue surface expression. And over time, they've developed a number of drugs that they then can combine for precision medicine. And I think this is one of like the great successes of the last 5 or 10 years. But CFTR only really affects 70,000 primarily European ancestry people. And while it's a huge health burden and a huge cost burden, it makes sense that it was solved. Billions of dollars went into solving this disease, and billions of dollars have been made on it. These are really shitty calculations based on Google searches, but roughly 350 million people have rare disease. I'm sure that's wrong, so you can tell me that. And around 8,000 of our genes are membrane proteins. So that's over 100 million other genes worldwide or people. Sorry, 140 million people worldwide who have rare genetic diseases and membrane proteins for which they can't be diagnosed right now for the most part, and there's no real path to developing targeted therapeutics. And I'd say if we can mechanistically understand how variants change sequence functions in membrane proteins and what those mechanisms are, and predict that, maybe we could make a dent on this. And so the real problem as a field for membrane protein people is that really most of the approaches in membrane protein biology are really hard biophysical approaches that has limited the development of general models and means that the field tends to be about a decade behind a lot of other protein fields. On the flip side, we have a handful of genetics assays, some of which I've developed, which usually aren't mechanistic enough. And so my solution has been to try and develop mechanistic mutational scanning, or really multi-phenotypic screening that allows us to understand how mutations affect membrane proteins. What I mean by that is pretty simple. Other people have been doing this as well for a bit, but it's kind of the idea that if we just focus on a single phenotype in our screens, like cell growth, then we're going to miss out on the translation effects or the folding effects or yada, 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 or all the different nested phenotypes or properties of proteins that are really essential for that protein to play its role in physiology. And so what we tried to do is we tried to develop multiple different readouts in my group that allow us to solve this problem. And before we get that, I'll just go over how we usually generate our libraries. We use this approach called Dimple that we developed. We made a cute little cartoon, so it makes us happy. Basic idea is this is a Golden Gate assembly that's done with oligo pools from Twist or Agilent that you can order and stitch in. We have open source wet lab code on protocols.io that we were doing before it was cool. No, I'm just kidding. And we have a GUI, and it generates primers for you. It generates oligos, and people are using it around the community, and it makes me really happy to know this helps other people. And a fun little feature is that we really allow also insertions and deletions alongside missense mutations, which I just think is cool and fun, and you should too, because who knows what's going to happen? We don't have enough of that data. And then we take those libraries that we generate with Dimple, and we make stable cell lines with Kenny or Doug's landing pad cells, and then we apply multiple phenotypic assays to this. Usually one for protein abundance, sometimes surface expression, sometimes specific activity, sometimes multiple assays for these. We use a lot of FACS for this, sometimes survival screens, and you sequence out, you get a heat map. We all know the drill. We've done this on a channel, a transporter, and a GPCR, and I'm not going to talk much about these two, the CUR 2.1 or OCT1, the ones on the furthest side, because these are all published as of like 2 weeks ago. One of them's published, so you can read the paper. And instead, I'm going to talk about this new data set on GPR68 that we just put out a preprint on that I'm really excited about. So GPCRs help us to sense the world around us. They're really professional sensors that allow us to perceive the world. And there's about 800 or so of them encoded in our genome, so they're the largest protein family. And they have quite distinct properties, but some of them, like the proton sensors here, can sense a strange phenotype or pH. And so pH is a very distinct phenotype from, say, a small molecule ligand because it's a proton. It's a physical phenomenon. It's not just like a ligand that binds into a pocket. And so these pH sensors are throughout our body in different places, such as in our bloodstreams, to sense CO2 level. And there's 3 of them that can sense basic pH, so GPR4, which is this leftmost one, then GPR5, which titrates around pH 7.4, physiological pH, or an acidic one, GPR68. And our collaborator, Ashish Manglik, was trying to answer this question, which is what is the molecular basis of proton activation? And he was trying to do this before I even started at UCSF. And by I mean, when I say he was trying to do it, I really mean his grad student, Nick, who is a biophysics graduate student, with support from some folks at UNC, XB Wong, who had this like giant screening core for doing dose response curves across tons and tons of conditions. And so we were trying, or not we, they really, we're trying to answer this question. And again, there's different GPCRs that sense different pHs. And they did a very simple, beautiful chimera experiment. I love chimera experiments because they sometimes work. And in this case, they tried to take a basic sensing pH, and then they added parts of an acidic sensing pH receptor, and they just tried to ask the question, when can it become an acidic pH one? So I'm having trouble with this thing, so I was like going to try and point, but I won't. So the top one is basic pH, and then they added one loop, then another loop, then another loop, and the unsatisfying answer is you needed the whole receptor to become an acidic receptor. So they're like, shit. But then they still went forward and they did a Herculean effort of solving a ton of structures, or they did this in parallel. And you know, they got structures solved in each of these states, which if you don't know, cryo-EM is really hard. Getting enough protein is really hard. And so these are the first structures that were solved. And they compared these, as one does, to classical GPCRs like beta-2AR, and they looked the same mostly, except for in a switch motif, instead of an N, there was a D, which maybe hints at something acidic. If you look at the orthosteric pocket where a ligand usually binds, there's actually a bunch of charged residues, which really hints at something, you know, pH related, but like it hints. There's a lot of charged residues on the extracellular face. So there's histidines and, you know, lysines on the positive sensing, or the basic one, and you know, Ds and Es on the other one, on the acidic sensing one. But they made hundreds of mutations in full dose response curves trying to answer this question and really struggled. And it's because they didn't have an unbiased map, right? And you know, it would be really great if we could generate unbiased maps of function or something. You know, what if there's a method to do that? So we had been working on, at the time, a way to measure GPCR activation in a FACS assay. So similar to what Shri Kasuri's group did and what Octon was trying to do when they started, and some other folks had been trying to do. And this is where we started. And by we, I mean intrepid rotation student Matt took these constructs I made and tested them. So this is DMSO, you know, this is a full agonist. You want these to look different. They didn't, so it's really sad. And this is an inverse agonist down here. And so compared to before, where they're using transcriptional readouts, we were trying to use a fluorescent readout, where you activate the receptor, you get cyclic AMP, and then you can read that out transcriptionally. We went to our friends in Utah, Justin English and Adam Zam, who are actually working with Octant to try and find new transcriptional response elements. They gave us some new transcriptional response elements. Matt foolishly joined my lab, and in a couple months got this to work effectively. He added a couple extra bells and whistles too. So now, you know, DMSO is here. Allopurinol is a neutral agonist, is known to have slight partial agonist behavior. A full agonist is much stronger, and an inverse agonist goes in the opposite direction. And this had been a general challenge for the field for a long time, is getting transcriptional readouts that were sensitive enough to detect the differences between different ligands for GPCRs. So then we were able to test this assay with the proton sensing receptor, and we were able to get really strongly different data. We can do full dose response curves and fun things like that. So we did a mutational scan with this, where we made, with Dimple, you know, full library. We included insertions and deletions for fun. We made stable cell lines using the HEC-293 landing pad cells. We did sort-seq. And, you know, we get this heat map. Very rich data set. Not going to spend a ton of time on it. It's hard to look at heat maps. Besides to say, transmembrane's bad. But also the other problem, of course, is that most mutations are going to be altered. Deleterious mutations are going to be acting upon folding and surface expression and not necessarily on activation. We needed to remove that. So we did a secondary screen using a fluorescent antibody. We then fit the data across these two different conditions, you know, just drew a fancy diagonal line from the synonymous variants and just did the Euclidean distance from there. and then we get this heat map where we've removed away a lot of the noise. Again, I'm not going to spend a ton of time on it because it's a heat map. It's very rich. We can map that onto the structure, and now we have an unbiased map of proton sensing. And whereas before they really struggled to pinpoint which residues were important, we now just have this unbiased map. You can look at the top of the receptor. There's about 9 histidines. Of them, 3 are important: H20, H269, and H175. We can look at that electrostatic pocket, and now it's very clear that the bottom of the pocket is actually really functionally important. And we find this network of residues that include an E and R, 2 tyrosines that seem to form the top coming down from a loop and really cluster around H269 and H20. And if you take H269 and you make a basic mutation to it, it becomes more active at basic pHs, and a positive mutation, a lysine, and a D goes in the opposite direction. And the heat map shows the same thing if I was to show it to you. So that's like super exciting. We could find a dosable, titratable residue. There's a lot of really other exciting things, like the E seems to change the Hill coefficient and things like that. And in fact, they had already done all the validation mutations in their ridiculous mutational efforts, so we just were able to then pinpoint them and make sense of it. So we have this sensitive transcriptional readout, which we can now use to do GPCR pharmacology. We have all these cryome structures for the first time that Ashish's group generated. We have a complete activation DMS. And with it, we can start doing integrated, unbiased structure function studies. And I think this will be really useful in GPCR pharmacology and structural biology, where there's a lot of fundamental questions left. But, we want to take a step back and think about what are all the different ways a variant can affect a receptor and affect a GPCR, from the folding effects to the trafficking, internalization, because that's a very important form of regulation in receptors, and of course, signaling. And variants within these receptors can change whether you have drug response or not, or how a drug responds. And this paper from Madan Babu did a very nice comparison where they asked the question of, for each receptor, what is the mutational burden on the x-axis? And then the y-axis is what is the number of FDA-approved drugs for each receptor. And they found the opioid receptor is one of the top receptors for mutations and drugs that exist for them. And so we tried to develop different assays or use assays that we had in hand. So we used a VAMP-seq-like approach. We developed a surface expression and internalization assay that's based on a halo tag that we have permeable and impermeable dyes that we can use. And then we used our GPCR signaling assay. This is now a GI inhibitory receptor, so we have to turn on basally the signaling pathway, and then we inhibit with the receptor, and we can get dosable behavior. So we did this ridiculous DMS experiment now. We have abundant surface internalization without ligand, and then at 5 concentrations with ligand. And so then we can look and see across these conditions what's the activation profile. So this is looking at without ligand, with ligand, more and more and more and more, and you can see this kind of behavior over time. We can look at specific residues and identify the residues that are important for function, such as the ligand binding pocket. They come out really strongly if we take the loss of function for function specifically. We can look at dose-response behavior across the conditions. So this is a position that's non-essential for function versus one that's really important for function, where G proteins bind. And we find some really weird ones that have more complex effects, where you can directly see, this is like literally just the raw DMS data, like using mean bin, none of the fancy statistics that we're in the process of developing with Harold and Jingyo and Jerome and the people who are smarter than us. But I think this is really exciting. We can do things like identify unambiguously an allosteric pocket or a site that's basally turns on the receptor when you make a mutation without a ligand, which is quite surprising, and maybe you could design a drug against that. So now what we've been trying to do, we're going back and doing some lower concentrations in the next couple weeks. We're also doing, multiple ligands, very similar to what Connor showed, but now with the GPCR signaling assay, where this is our assay data. So this is like theory of what pharmacology should look like, and this is our facts data. I can't express enough how amazing it is that we can see the difference between a partial agonist and full agonist, and we can start to ask fundamental questions about that. I'm super stoked. We can also learn basic things about biogenesis, like which parts are important in biogenesis. This came out last night at dinner where people were asking me like, or like I think someone said like, oh, what if we could just like make mutational scans across representative proteins? And I was like, well, we've done a number of these. And we started to compare them. And to our surprise, this is the mu-opioid receptor, TMs 2 through 4 are important. This is GPR68. All the transmembrane domains are important. This is GPR161, which is orphan receptor, and like where the ligand binding pocket usually is becomes important for folding. That's really weird. I don't have an explanation besides proteins are weird, and if they share the same fold, they don't necessarily fold the same way, which is exciting, and maybe with more data we can get there. We've developed a number of these data sets, generated quite a few. I showed you very briefly, we did this within a transporter, we did this in the GPCR, we did this in the potassium channel, and we find overall most deleterious variants affect biogenesis or folding. We find that not all transmembrane domain residues are equivalently sensitive. So you can't just be like, oh, it's a transmembrane and it's like facing the membrane. That's a sufficient rule. And we find the non-transmembrane folded domains or the cytosolic or extracellular domains are actually really important. This is known from CFTR that if you mess up the nucleotide binding domains folding, you mess up the whole folding. Maybe they act as like co-translational chaperones or something. That's where we are in what we know. I think the first point actually is pretty big impact. Because while we're trying to answer these questions of what are all the mechanisms that mutations can impact a membrane protein, we've learned it's mostly biogenesis. And so how can we start to address these problems broadly, and how can we do this as a field? And I would love help from everyone if you want to play together, because I love collaboration, and this problem is way bigger than me. But I think we need ways to make combinatorial libraries and to do that efficiently. I think this is one of the biggest problems for us to study proteins. I think we need better mechanistic phenotypic readouts. I think we've gotten somewhere, but we need more of them so we can really answer questions of how variants are affecting proteins. We need a lot of high-quality data sets of how residues are interacting, because I think that's the gap in machine learning oftentimes, is we don't have these mechanistic higher-order models or data to model. We need to know how proteins are actually interacting, other proteins are interacting with our proteins. And we need new statistical frameworks. And for that, I can thank Jinyo, who you all got to see her brilliance yesterday, and Harold and people like that. With that, I want to thank you. Thank for listening to me ramble. Thank my lab, who are currently in a competition to make the coolest, most artsy picture of the lab. And our collaborators and funding. Thanks. Oh, and Priyanka is a postdoc who's co-mentored by me, who's going to be giving a poster later on something completely different. So go check that out too. Yeah.
Thanks very much for those. Okay. I'm sure we're going to have some questions. Actually, I was quite interested in the variant that gives the baseline activation of the...
Sorry.
That was a joke, which I'll tell you later. Okay, questions? Got one there. We've got one down there.
Hi, great talk. Laura Haynes, Michigan. I was wondering, have you done any screens looking at variations in the lipid environment that the proteins are in, or?
I think that would be really fun. It's hard to know how you actually perturb that, but yes, I would, or no, I haven't, but yes, I would like to.
Hey, I'm Jordan from the Broad Institute, and I'm wondering, are you doing anything from the machine learning side to increase the accuracy of the MAVE readouts and to kind of,
Whether we're doing anything on the machine learning side to improve the data quality?
Sorry, not on the machine learning side.
Oh.
Are you doing anything to increase the fidelity of like the assay readouts? So like you could have more significant figures or more granular bins. Reading off of the.
So we're now doing like full dose response curves, right? I mean, I showed that, and that's with that goal in mind. So the short answer is I'm super happy to talk about it. But we're always trying to think about new phenotypic assays that are more sensitive. We don't try and do more bins because we already have a ridiculous number of conditions that we're trying to do, and that would be even more ridiculous. So, yeah.
Thank you.
Okay, I'm going to do an abusive interrupt here again. So given what you said at the beginning of this, how the world's going to be taken over by computational methods and everything, so have you tried the very simple thing, given that computational methods are likely to be convolutions of many different aspects of function, or nested phenotypes, as somebody said, and then subtracted the, presumably you've played with doing the predictions and then seeing what is wrong with one of the assays?
Yeah, yeah, absolutely.
And it doesn't work?
Yeah, yeah, yeah. So we tried that. I did that with the transporter data, and the transporter data actually behaved really well in like alpha missense, and in that, so.
Okay.
But
We can talk later. It wasn't quite what I was meaning.
Sorry.
It's okay. We had one more question, if we have got time, if that's okay with...
I thought you stole my time, Debbie. Okay.
I stole your question.
So no. So I was just very curious. Like, how do you think like the mutations just biophysically might be controlling the Hill coefficient?
So in that case, you have like a network, right? And I just think it's breaking the network. So there's a lot of cooperativity in this receptor. There's a bunch of these histidines, and I think that there's coupling across these residues, and then it's very likely it's just breaking the coupling.
And does it seem like it's controlling the Hill coefficient independent of like the pH at which it turns on?
Not in that case, maybe. But like, we don't have the common, like what you would really want is a combinatorial library in those sites, and then to feel really confident in that. It's more like we have the dose response behavior from the from like other assays, and we can stare at them. But that's what we want to get at in like more conventional systems. We had a lot of problems with the proton sensor, partially because we didn't know the answer already, which is like cool, but really hard when you have this really rich data set. So.
Okay, thanks.
Okay, well, thanks, Willow. I think in the interest of time, we'll have to stop. Thank you again. And